Okay, thanks so much, everybody. Thanks for sticking around until the end of the session here. I'm going to be talking about some recent work from my group on a method called phylogenized 2. And the purpose of this is to try to identify microbial genes that are associated with particular host phenotypes. This will be a little preview of maybe some of the talks in the microbiology session, but we are kind of walking culture vessels for microbes, and these microbial communities influence our health in a variety of ways, some of which I'm showing here. And in some of these cases, we can even transfer the phenotype by transferring microbes. So for example, in using something like fecal microbiome transplant. So this is great. But just knowing about these associations is something that we want to move beyond. And the reason is that even when we know there is some kind of causal association, we don't always understand why. And because of that, the tools at our disposal for intervening are kind of blunt instruments, right? We can think about things like antibiotic treatment, which really clears out a microbial community. Or we could think about something like, as I mentioned, fecal microbiome transplant, which is a very effective treatment for diseases like Clostridium difficile, but that involves taking an entire community from one person and putting it into another, right? And it's not risk-free. People have died from microbiome transplants that were inappropriately screened, for instance. So we would really like to understand what we're doing better to design more specific treatments. So for example, if we know what genes are involved in a particular, giving rise to a particular host level phenotype, we could imagine choosing different strains of microbes that have those specific genes, or we could even imagine for genetically tractable microorganisms, engineering new probiotics. Or we could imagine if we have a gene, that means that we potentially have a new drug target. So we could also maybe design drugs that target really specific bacterial proteins instead of things that are really broad spectrum. Okay, and so I'm going to be telling you about one effort that's sort of ongoing in my group to develop a tool that can help get us from these associations to something that's at the gene level. And so I'll start by showing you a pretty typical result that you might get from a case control study in the microbiome. So let's say that you do metagenomic sequencing on a bunch of cases of people who have a certain disease, as well as people who are healthy controls. And then you look for the taxa that are enriched in each population. And so this is great, but it sort of leaves the question of how do you interpret these results? So one way to interpret them is by gene content. And so here I'm showing you two possible candidate genes, G1 and G2. And you can see that these are kind of both appear to be decent explanations for this enrichment. But there is a problem with this, and it's one that you may be able to guess based on the theme of this session, and that's that these species are not independent, right? They're related by vertical descent. And so closely related species tend to share both genes as well as phenotypes. So this is a problem when we're doing these kinds of large scale analyses. And unlike Nathan and Maria, I didn't invent a method to deal with this. I just appropriated one from the literature. So it turns out that there is already a solution for dealing with this type of data. It's a big class of methods called phylogenetic comparative methods that Felsenstein and Graffin really sort of invented in the 80s and 90s. And they're very commonly used in kind of like macroecology, but I think they're very underused in microbial ecology. And particular in kind of like case control studies of the human microbiome. And the trick with these methods is that they are kind of like forms of regression, but they assume that species covary in their phenotypes proportionally to how recently they diverge. So things that diverged very recently are assumed to share a lot of variation. Things that diverged a long time ago are assumed to be more independent, right? And so because these tools are kind of underutilized and we think that they give a lot of really interesting insights into these data, we developed a tool called Phylogenize, which is supposed to allow other researchers to apply these methods to their own data. And this is a very complex slide, so I'll just give you the highlights here. Basically, the users would provide either shotgun data, mapped shotgun data, or 16S data, and then Phylogenize kind of takes care of the rest. So we provide the estimates of what genes are in the pangenomes, we provide the phylogenetic trees relating to species, and we also estimate our kind of measure of enrichment, which in this case is differential prevalence. I'll get back to that in a couple of slides. And then we report these the associations that we find by fitting these models. And so as we continue to use Phylogenize in new settings, we discovered some shortcomings that needed to be addressed. And the first one of these is database coverage. So Phylogenize used at this point kind of an old, an older version of RefSeq to get gene content estimates. And this is a figure from a paper. Sorry, I don't have the citation there, but this is NAFAC et al. And what they're showing here is the proportion of a metagenome that actually maps to something in our databases. And you can see that if you're studying human skin, like great news, you're doing pretty well. But once you start to look at, for example, the human gut using stool samples, you're doing much worse, particularly outside of the kind of global north, right? And one particularly shocking thing is that if you look at how much of a lab mouse metagenome actually maps to databases, it's actually very little. It's around like under 10% on average, right? And that's a huge problem because we use lab mice a lot in studies of the microbiome, right? And in fact, when we tried to apply Phylogenize to some mouse data, we found that we had basically no signal. And so our solution to this is to include more modern databases that also include a lot of metagenomic assembly. And so this work is really being pushed forward by a bioinformatician in my group, Kat Kananen, and Nia Tran, who's a graduate student in my group, who's just started working on this project. And basically, we're including two large genome collections, one of which is human gut focused, that's UHGG, and then one that covers a much broader range of environments. And so we're using this pretty recent revision of GTVB. Some of this work was already done for us. So we're piggybacking off some work that was done for this other tool, MIDAS2, of clustering these, constructing pangenomes from all of these individual genomes. And so what we're doing is annotating these genes into protein families, right? And so that's the next step that you need in order to apply Phylogenize to these genome databases. Another issue with the first version of Phylogenize that I mentioned, you might recall that I said we were looking at prevalence and differential prevalence between conditions, right? And so this is enough to see very, qualitative changes between environments. So for example, if you're comparing the gut to the oral cavity, you can see a lot of differences, right? But it doesn't work as well for looking at, for example, the same environment in two different conditions. So looking at the gut in health versus disease, for example. And the reason that we used prevalence initially is basically because the math is simpler, and because we need estimates of a phenotype that have two properties, right? We need to be able to account for how confidently that phenotype is measured, right? So we don't want to get fooled by cases where, for example, we see a microbe once in cases and twice in controls, and we conclude it's like 100% greater in the controls, right? And so what we do is we want to shrink these estimates before we put them into the phylogenetic models. And so previously, we had kind of rolled our own solution to do that with prevalence. And then the other issue is that we wanted to be robust to, if you remember from Justin's talk the other day, these kind of compositionality effects. And basically what this means is that when you are sampling a microbiome through sequencing, you lose the information about the absolute quantity of cells that were in your environment, right? You only have relative abundances. And this can give rise to a lot of like spurious effects that can really distort the results of these analyses. So we needed something that we could, so we needed basically to be able to shrink our estimates of the phenotype of interest, and we needed a phenotype that was robust to these compositionality artifacts. And so the solution that we came up with is to use some of these modern differential abundance methods. Right now, we're using a tool called GenCompBio. And the important thing is that this reports both an estimate of log ratio change or like effect size, but also the standard error around that estimate, right? And so you can, in principle, use any tool that reports both of those things. And then we feed them into a kind of more general purpose shrinkage method called adaptive shrinkage. And this gives us a shrunken measure of the differential abundance that we can then plug into these phylogenetic models. And so in the last couple of minutes, I want to take you through some real results. And again, this work is really being pushed forward by Kat, as well as two graduate students in my lab, Nia and Stephanie. And this work focuses on the commensal Clostridia in liver cirrhosis. And in particular, it focuses on this family of bacteria called Lactospiraceae, which my lab is very interested in. In general, for those who don't know, gut microbes are dominated by two different phyla, the Bacteroidetes and the Firmicutes. And one of the most common Firmicute families, so these are gram-positives, right, are these Lactospiraceae. So they're very, very common in healthy people. And what's interesting is that there are a lot of reports of the Lactospiraceae being depleted, specifically in liver disease, right? And so there are multiple independent observations of this. And also there's even some results from mouse work that in a different type of liver disease, restoring the Lactospiraceae, so giving mice these bacteria, can also help rescue some of the disease phenotype. So what we found when we started to look at this is that actually not all of the Lactospiraceae are as depleted in cirrhosis. And so this is showing these moderated estimates of differential abundance across the tree of Lactospiraceae, right? And you can see that some of them are quite depleted, but others seem to be not depleted or even enriched, although again, because of compositionality, we don't know exactly, it may be just that they're depleted less, right? But in any case, there's a big difference when you look species by species. And so this makes it sort of a prime candidate for analysis with phylogenize. And so we actually also compared to both just a standard non-phylogenetic corrected linear model, as well as another tool that's recently been published. I don't have time to get into how that works. But what we found is that we recovered several significantly associated genes. So in this case, we recovered like 37 positively associated, so associated with the case. If you compare that to the linear model finds tons of associations, but most of these appear to be spurious, right? Most of these appear to just be essentially clade markers, and I'll show you an example of that on the next slide. And when we compared to this other method, POMS, it seems that POMS didn't really recover any significant hits at a more restrictive false discovery rate threshold. And even if we loosen up this threshold, it doesn't seem to recover as much as phylogenize. So we think this is because we're sort of balancing sensitivity with specificity here. And I'll show you some examples of our top hits. So on the left is one of our top 20 hits from phylogenize. And what you can see is that on the left here we have, on the left we have the change in abundance, so this moderated change in abundance. And then on the right, you can see gene presence absence. And you can see that kind of like across the whole tree of the Lactospiraceae, right? Gene presence and absence is correlated with higher abundance. And when you look in contrast at some of the top hits from the linear model, right, which is on the right side here, you can see that these hits are really phylogenetically restricted, right? And so that's what I'm talking about when I say that the linear model tends to pull back like clade markers, right? And so we were very interested in what some of these hits were. One of them is this gene RNR. And actually, this is an aerobic ribonucleotide reductase. And we found this to be really odd because Lactospiraceae are obligate anaerobes. They're very sensitive to oxygen. So when we started to look into this, we found that somebody had already reported that in a totally different phylum that also had an aerobic ribonucleotide reductase, this gene was shown to increase their tolerance to transient oxygen exposure. And one of the things that we know is that gut inflammation can increase reactive oxygen species, reactive nitrogen species, and potentially also more oxygen can leak into the lumen of the gut. And so this may explain why these Lactobacilli are better able to persist in this environment. For the sake of time, I'm going to skip over one of our other hits, but there was also an association with the anaerobic oxidative stress response. So what I want to leave you with is that we have an updated version of this tool, which will be released soon. If you want to apply it on your own data, you can sign up here to see when it is going to become available. We think that this really gives us a lot of power, but also a lot of specificity when it comes to trying to find microbial genes that explain these patterns that we see in, for example, health versus disease. Okay, and so I want to thank the rest of my lab. Kat and Nia both have posters. Kat gave hers yesterday. Nia is giving hers today. And then I also want to thank the rest of my lab. We also are a wet lab, so we do a lot of wet lab microbiology, which I didn't have time to tell you about, but happy to talk about that later if you're interested. And then I want to thank my funding. Thanks so much.
Okay, thank you, Patrick. I think we'll take one question before we head off to lunch. Anybody? Sure, go for it.
Yeah, the first version of it is, oh, sorry. Yes, so the question was, is this tool available? We're expecting the second version to be available later this summer. The first version is available. It's on GitHub for the lab GitHub. Yeah. And it's available also, you can access it as a package for QIIME 2.